First of all, let me express my gratitude to respected Professor P.K. Shashidharan, the well-known medical teacher and social health activist, for inaugurating this CME series with an introductory talk on vitamin D last week. Special thanks to Dr. George Jacob, AKMG Emirates President, Professor G.S. Patnaik from Bhubaneswar, and Dr. Subbu Koshi, AKMG Emirates Secretary, for gracing the occasion during the first part of this series. Today, we will have the second part of the series, that is with vitamin D in health. Today's chairpersons are Dr. R.C. Srikumar, who is the chairman of IMA Research Cell, and Dr. Jamaluddin, CME chair, AKMG Emirates. The role of nutrients and molecular biochemistry of this very essential life-sustaining pro-hormones. Now, after listening to the talk, please do post your queries in the comment box of that. I am permitted
the participants list
after the talk. The rest will be covered during the additional video on vitamin D in health and disease after the 3 talks are over. I request everyone to mute yourself so that the talks are not disturbed. Thank you. Please do post questions related to today's topic only. This is a special request because the dosage schedule and the connection between vitamin D and diseases will be covered in detail in part 3 of the talk next Friday, same time. So you can either post in the chat box, this chat room here, or below the comment boxes on the Facebook Live. Thank you. Now, today we are very lucky to have 2 eminent doctors to chair this session. First, I am privileged to welcome Dr. R.C. Srikumar. Professor Srikumar is the chairman of IMA Research Cell and Digital IMA, Indian Medical Association, Kerala State. At present, he is the additional professor of surgery and head vascular division, Medical College Hospital, Thiruvananthapuram. He has held many responsible posts in IMA as well as Vascular Society of India and Association of Surgeons and other professional and social bodies. Welcome, Dr. Srikumar, and special thanks for accepting my invitation. I would request Dr. Srikumar to deliver his opening remarks. Dr. Sri.
Respected Dr. George Jacob, President of AKMG Dubai, and my dear friend Dr. Hanish, and dear friends. I'm very happy to have with you for 2 reasons. One is vitamin D is a very important topic of today, and to introduce Dr. Hanish, who is a close friend of me and Indian Medical Association, and for the last 1 year he was the editor of the IMA Live, that is the digital platform of public health of Indian Medical Association. He has done excellent job as the editor, and he has published a lot of articles in IMA Live. So, and vitamin D, of course, is one of the is often depicted as the vitamin of the century. Still, many of the clinicians, including us, have very limited knowledge about vitamin D. We need to have an extensive knowledge of vitamin D, and that will help us for day-to-day management of our patients. So, in this scenario, this lecture is very, very important, and I hope this will benefit many of our clinicians and our patients. So, with pleasure and utmost privilege, I am moderating Dr. Hanish Babu's lecture on vitamin D. So, thank you for inviting me for this wonderful program. Thank you very much.
Now, our second chairman is Dr. Jamaluddin Abubakar. He is a leading pediatrician in these parts of the world, and Dr. Jamal is the CME chair of the Association of Kerala Medical and Dental Graduates, UAE chapter. He is also the medical director of Metro Medical Center in Ajman, UAE. He is a very active health educationalist in the media. Not a day goes by without seeing Dr. Jamal either on the TV or listening him on the radio, like our president, Dr. George Jacob. They both are, I think, competing with each other to be on the media. They are doing wonderful work for the public health arena. Welcome, Dr. Jamal. Please do deliver your opening remarks. Dr. Jamal.
Thank you, Dr. Hanish. Thank you, Dr. Hanish, for having me in this wonderful presentation. I listened to your last presentation. It was viewed by a lot of people, and it was we know that it is so extensively researched and presented, very well presented talk. As we all know that vitamin D plays an important role in the calcium and phosphorus metabolism and in the bone mineralization, but it has gained a lot of attention these days as emerging evidence that it is associated with autoimmune and inflammatory disorders, cardiovascular risk, metabolic syndrome, malignancy, and a lot of in almost all the diseases in our body. So it's something beyond what we see as vitamin, and we are looking forward hearing from you more. Thank you, and all the best.
Thank you, Jamal. Now, first, before starting the talk, let me declare that I have no financial or other conflict of interest in presenting this talk and this series, except that of public interest in mind, of course. In this part of the talk on vitamin D in health, we will discuss the following in brief: history of vitamin D, overview of vitamin D metabolism in our body, and functions of vitamin D in health. We will also explore how vitamin D functions at molecular level and its mode of action in a little detail, with more emphasis on its role in maintaining an optimum level of immunity. A little history before we move on to the science of vitamin D. History of vitamin D is also the history of all vitamins. I will try to make it as short as possible. It's a vast subject in itself. Vitamin D, as you all know, is an ancient hormonal molecule. It is photosynthesized in all forms of life, almost from the phytoplanktons of 750 million years ago to the present day mammals and humans. It can be considered as one of the first natural antioxidant pro-hormone and one of the first pro-survival agents in mammals and humans. The Nobel Prize for Chemistry in 1928 was awarded to Adolf Windaus for his studies on the constitution of the sterols and their connection with vitamins. Actually, he was the first person to receive an award mentioning vitamins. As you all know, rickets, scurvy, beriberi, and other such diseases were known for millennium. The Roman description of rickets goes back to 2nd century AD. German chemists in the 19th century, led by von Liebig, put forth the dogma that an adequate diet consists of 12% protein, 5% mineral, and 10 to 30% of fat, and the remainder as carbohydrate. This was proven wrong by these researchers who fed the recommended portions of these purified dietary components to animals and found that they failed to thrive and survive. It was in 1897 that Ekman experimented by giving unpolished rice to prisoners who were previously fed predominantly a diet of polished rice. This solved their beriberi problem, which was very prevalent among the prisoners at that time. Unfortunately, Ekman concluded that the polished rice possessed a toxin that was neutralized by a substance in the hulls of the rice, the outer capsule of the rice. Later, Grins, that was in 1901, he was a colleague of Ekman. He revisited the question and correctly demonstrated that hulls contained an important and required nutrient that prevented beriberi. Still, at that time, the idea of a vitamin or a nutrient, essential nutrient factor had not yet been given birth. The 18th century sailors were aware that eating liver cures night blindness. lemon cures scurvy, and cod liver oil is the best cure for rickets. It was Hopkins in 1906 who first postulated the existence of an essential dietary factor necessary for the prevention of diseases such as scurvy or rickets. Casimir Funk, he was the person who coined the name vitamin. Actually, he coined it as vitamins, because he thought the vitamins were containing an amino acid. So to name it as vital amine, he named it vitamin. But later it was found that vitamins were not amine compounds. The E was dropped from it by Drummond, and they began to be called by vitamin. That was in 1920. At present, there are 13 vitamins which have been found, which have been discovered between 1913 and 1948. Now comes the era of vitamin D. Mukhathala and co. in 1913 and 14 found that a fat-soluble factor A prevented serophthalmia and stimulated growth in rats. Mukhathala in 1916 and Osborne and Mendel in 1917, almost simultaneously, though they were apart by in different continents. They almost simultaneously found out that it was a water-soluble factor which prevented beriberi, and later it was named vitamin B1. Mellanby in 1919 put forth a hypothesis that the cause for rickets was a diminished intake of an anti-rachitic factor, which is either Mukhathala's fat-soluble factor A or something similar. And it was found in abundance in butterfat and cod liver oil. So he thought it could be same. Drummond in 1920 again coined the word vitamin and named the factor A as vitamin A, the water-soluble factor that worked against beriberi as vitamin B1. And by that time, vitamin C was also discovered, though its chemical composition will be discovered quite later on. And A, B, C were already named. So by this time, Mukhathala again, he revisited the question of anti-rachitic factor, and he did a very intelligent experiment. He bubbled oxygen through the cod liver oil, which destroyed vitamin A, and found that this preparation was no longer able to prevent serophthalmia and vitamin A deficiency, but it still retained the ability to cure rickets. So he rightly concluded that the factor that cures rickets is a new vitamin, and they called it vitamin D. So this is how vitamin D came into this thing. You can imagine in 1926, Mukhathala and Nina Simons wrote this excellent review. A balanced diet is the only safe diet. And it is unfortunate that even after almost 100 years, still the mankind is not strictly following this. Actually, Mukhathala should have received a Nobel Prize for his contributions in discovering vitamin A, vitamin B1, and vitamin D. But unfortunately, he did not. It was Adolf Windows who got the first Nobel Prize in 1928 for the sterols and vitamin D, and Albert St. Georgi, who isolated ascorbic acid in 1928. He got the Nobel Prize. This is the list of Nobel Prize winners for vitamins, their discoveries, their isolations, their synthesis, and for studying the structure of vitamins. Unfortunately, our Mukhathala is not there. Haldinsky, a physician in Vienna, and Chick et al. In England, found that suffering from rickets could be cured by exposing them to summer sunlight or artificially produced ultraviolet light. Hess and Unger also noted that sunlight could cure rickets. In 1935, The 7-dehydrocholesterol was isolated by Windows et al. And vitamin D3 was identified in 1937 by Windows and Bock. Holick et al. In 1977 provided evidence that pre-vitamin D3 is formed in the skin on radiation. So the connection was slowly building up that not only the dietary sources, but vitamin D can be produced or synthesized in the skin from the ultraviolet rays. Vitamin D became so popular in the 1930s and 40s, and a wide variety of foods and beverages, as well as even personal care products were fortified with vitamin D. You can see they included not only milk and other dairy products, but also soda, beer, hot dogs, custard, and even soap and shaving cream was fortified with vitamin D. But later, a few cases of vitamin D toxicity discouraged vitamin D fortification of dairy products in some countries. Along with this, the universal fear about sun-induced skin cancers reduced sun exposure. As a result, the rickets has staged a comeback in some countries. Sweden and Finland, even now, along with many other countries, permit milk to be fortified with vitamin D. It is worth noting that if the milk has been fortified with 100 international units of vitamin D or per 8 ounce of the 8 ounce, and for the past 80 years in most of the countries, there have been no reports of toxicity except for the earlier stages where it was suspected that the infants also received high dose vitamin supplements, not just the fortified milk. In the past 10 years, juice products, including orange juice, have been fortified in the United States with 100 international units of vitamin D per 8 ounce without any reports of toxicity whatsoever. A very interesting thing with the advent of COVID-19, this was a study carried out which supported that when the vitamin D fortification started in the 1930s, which went on till 1930 and gradually reduced in many countries because of a few reports of toxicity. During that period, there were no influenza epidemic and pandemic during that period. You can see straight away, this has been reported. Something that we should think about. In the last talk, we had already examined all these points and pointed towards some negative reports and told that when negative reports come about an essential nutrient, what we should do, we should go to the basics. That is what we are doing today. The term vitamin D is unfortunately an imprecise term. It is not actually a vitamin, as it can be produced in the body with adequate sun exposure. Nor is it a hormone, as it has dietary sources also. That is why it is well described as a prohormone, produced photochemically in the skin from the metabolite of cholesterol, that is the 7-dehydrocholesterol, in a physiologically inactive precursor state, which is later converted into a hormone in the active metabolite, as an active metabolite in the kidney and target organs. That is why it is called a prohormone. Vitamin D belongs to the family of fat-soluble vitamins, which include vitamin A, D, E, and K. This we all know. These vitamins are absorbed well with fat and are stored in the liver and fatty tissues. The actions of vitamin, as we have already seen, are similar to hormones, as it controls cellular functions at a molecular level. This is what we will see today. There are two main forms of vitamin D in the diet. Vitamin D2 or ergocalciferol, which comes from plant foods, very rare and mostly found in mushrooms. That also mushrooms which are grown in sunlight. Vitamin D3 or cholecalciferol, which is of animal origin. We get lots of cholecalciferol from fatty fish like salmon, sardine, and also from egg yolks and liver, especially beef liver, are rich sources of vitamin D3. However, as we all know, sunlight is the best natural source of vitamin D3. The UV rays from sunlight convert, as we have seen, cholesterol in our skin into vitamin D3. The classical functions of vitamin D, we know it involves the mineral balance and skeletal maintenance. It has been known for many years. But with the discovery of vitamin D receptors in various tissues, several other biological functions of vitamin D are increasingly recognized, and its role in many human diseases like cancer, diabetes, hypertension, cardiovascular and autoimmune, and dermatological diseases being extensively explored. There are both pro and con reports on this. We will examine all those extensively in the next talk. Here we will go into the molecular basis of the vitamin D mechanism of action as well as the metabolism of vitamin D. The wide variety of non-classical functions of vitamin D involves almost many of the cellular functions like regulation of cellular proliferation, differentiation of cells, apoptosis, regulation of innate and adaptive immunity. In this review, we will discuss and summarize the latest findings on the non-classical functions of vitamin D at the cellular and molecular level and its role in complex human diseases. This is the metabolism of vitamin D. You can see a very important slide. You can see the sun's UVB rays of the sun acting on the metabolite of cholesterol, that is the 7-dehydrocholesterol. It is converted to pre-vitamin D3. It is called pre-vitamin D3. This is called 7-dehydrocholesterol is called pro-vitamin D3, which is acted upon by absorption of 1 photon of ultraviolet B from the sunlight. And remember, UVB is most prevalent from 10 a.m. till 3 to 4 p.m. in the afternoon. Please remember this. Many times, many patients have come to me and told that their doctors advise them to go to sunlight to expose themselves during early morning and late evenings. No, they are not going to get UVB during that time. There may be a little, but not much. But of course, I tell them exposing to sunlight is always good. Anytime is good, but for you to get vitamin D, you have to expose between 10 a.m. till 3 to 4 p.m. Now, further conversion of this pre-vitamin D3 to D3, vitamin D3, is regulated by body heat. Please remember this, body heat is required. The process that takes place over several days. It takes several days. It is not a sudden reaction, maybe almost 4 to 7 days, even up to 2 weeks in some cases, when the body heat is not preserved, especially for us who are sitting in the AC rooms or for those who are living in temperate climates towards the Arctic and the Antarctic regions. For them, the conversion can take longer time. But of course, we will see in the next talk that the ethnic differences during the evolution has developed in such a way that many ethnic groups can survive with low vitamin D and low calcium. Their calcium metabolism is rerouted away from vitamin D3. Both vitamin D3 that is spontaneously formed in the skin and that comes from the digestive tract through intake of vitamin D containing food, especially the non-vegetarian food. They are carried to the liver by the vitamin D binding protein. And in the liver, the enzyme, it is a cytochrome P enzyme, that is the 25-hydroxylase. It converts vitamin D3 to 25-hydroxy vitamin D3, which is the major circulating vitamin D. This vitamin D, it's not only circulating, it is the major form of stored vitamin D as well. So this is the vitamin D that we usually test to see what are the levels of vitamin D. The 25-hydroxy vitamin D3. The total levels are seen. Now, these are taken to the liver, but still it is in an inactive form. This is only the circulating and the storage form of vitamin D. It is not an active, biologically active form. For it to become biologically active, it has to be converted into 1,25-dihydroxy vitamin D3, or well known as calcitriol. We'll be using the name calcitriol instead of always repeating this 1,25-dihydroxy vitamin D3 from now onwards. So 25-hydroxy vitamin D3 is converted into calcitriol by the action of 1-alpha hydroxylase enzyme. That is also cytochrome P enzyme. So this conversion makes it active metabolite, and this then acts through the vitamin D receptors. We will see how. Just we will, as it is a very important, we will just go through the steps in a little more detail. The important thing to remember is that before our body can use vitamin D, it must be activated through a series of steps, one in the liver and one in the kidney. So if there is a liver disease or there is a kidney disease, it is highly probable that the vitamin D may not be available for its actions. One. Second, this was thought to be very essential, but later on it was found that vitamin D is activated not only in the kidneys, it is also activated in each and every cell through the action of vitamin D receptors. Every cell contains vitamin D receptors. Once this vitamin D receptors from the kidneys, the vitamin D binding protein takes the vitamins to the vitamin D to the both the activated vitamin D as well as the non-active vitamin D, circulating vitamin D to the organs. And whenever there is stimulation or the cell is supposed to perform a function that is written in its DNA library for the genetic decoding, this inside the cell itself, the activation of vitamin D is taking place. This is very important in understanding the actual metabolism of vitamin D. Let us go a little deeper now. Now this calcium metabolism, as we all know, is modified by vitamin D by a negative feedback control system that includes the parathyroid hormone, bones, kidneys, and intestine. Serum calcium is kept within a very narrow range by parathyroid hormone and calcitriol by stimulating osteoclastic bone resorption, is in the bones, and the reabsorption of calcium in the renal tubules, and the intestinal absorption of calcium in the small intestines. These all three areas are controlled by vitamin D and parathyroid hormone. It is a negative feedback mechanism. Secretion of parathyroid hormone by the parathyroid glands is stimulated by serum phosphate, low serum phosphate, and hypomagnesemia, and inhibited by calcium. It is inhibited by the hypocalcemia and hypomagnesemia, and stimulated by the increased serum phosphatase. And this in turn increases the parathyroid hormone secretion, and these actions take place via the stimulation of the calcitriol. This is in short the vitamin D, calcium, parathyroid hormone, phosphate, and magnesium loop that really affects the bone mineralization and also the calcium metabolism inside the body. In short, the parathyroid hormone and calcitriol have a mutually inverse relationship. Interesting species differences are also there in the ability to synthesize vitamin D through the sunlight. The skin of humans, horses, pigs, rats, cattle, and sheep have adequate quantities of 7-dehydrocholesterol, which can effectively be converted to cholecalciferol. But in contrast, the skin of dogs and cats are different. It contains significantly lower quantities of 7-dehydrocholesterol than other species. So its photochemical conversion to cholecalciferol is quite inefficient. And dogs and cats thus appear to rely mainly on dietary intake of vitamin D more than do other animals. So next time, don't be surprised if your cat shows more interest in fish than anything else. It is a survival instinct. Some important points to note on vitamin D metabolism. The production of vitamin D in the skin is directly proportional to the exposure to sunlight and inversely proportional to the pigmentation of the skin. This is important. The higher the amount of melanin, the lower the amount of vitamin D produced in the skin. Excessive exposure to sunlight does not result in vitamin D deficiency, since excess provitamin D3 are destroyed by sunlight itself. This is also very important, because if we take supplements, higher dose of supplements, there is always a chance of vitamin D toxicity, but this never occurs through sunlight. There is natural mechanisms where sunlight can degrade the vitamin D. So, next is the when we have the choice of taking vitamin D3 and D2, sometimes the patients may ask, which is more effective? Of course, vitamin D3 is twice as effective as vitamin D2. So for deficient patients, always advise vitamin D3, sorry, rather than D2. Now, the major discovery, the recent discovery was about the vitamin D receptors in all cells of the body. Thousands of vitamin D receptor binding sites are present throughout our genome and controls hundreds of genes in our body. This is to be really kept in mind, because I have heard many people, including doctors, asking, what is so special about vitamin D? Is it just another snake oil? No. We have to realize that vitamin D is the only vitamin that can be produced from the sunlight. It is similar and equivalent to the photosynthesis that is being done in plants. And photosynthesis is the basis of life on this planet. So, you know what happens if we keep plants away from the sunlight in a dark room? What happens? Same thing can happen to us. Fortunately for us, we don't remain fully inside. We at least go out sometimes, and we also take some dietary intake which contains vitamin D. So please don't reduce or see vitamin D in a very, negative way. Take it in a positive way. Whatever these studies have come, we will examine them. There are some things that we cannot digest, because it is very difficult to in the last talk, those who listen to the last talk will remember the example of the machine and electricity. It is like this is an essential nutrient. How do we prove that this essential nutrient is not having any effect in any viral infection like COVID-19? How can we prove? When see, it is like same example I will tell you. It is like providing electricity to a machine. Imagine our body is a machine, and if it is running on a low voltage for a long time, you know what happens? The wear and tear, and gradually the machine will break down. Now, when the machine breaks down, if you give more electricity, will it work again? Will it get repaired? No. The same thing happens with vitamin D. When the nutritional deficiency, not only vitamin D, all nutrients, when the vitamin D or nutritional deficiency has occurred, and once the damage has been done, if you give more vitamin D or more nutrients, the damage may not be undone. It can prevent, of course, further damage, but cannot be undone. Then how can you infer at that time that vitamin D has got no effect in infections or COVID-19? No. We'll see why. This is why we have come to the basics. This we have already seen. Without vitamin D, especially in the calcium metabolism, only 10 to 15% of dietary calcium and about 60% of phosphorus are absorbed. Vitamin D sufficiency, of course, enhances calcium and phosphorus absorption by 30 to 40% and 80% respectively. Thus, a low blood level of vitamin D is linked to a greater risk of fractures. Vitamin D also prevents, as you all know, rickets in children and osteomalacia in adults. Together with calcium, vitamin D also helps protect older adults from osteoporosis. Now, how does vitamin D mediate its functions? We have seen vitamin D receptors are there in all the. How does it mediate the function of vitamin D? Vitamin D receptor is a member of nuclear hormone receptor family, which includes many of the hormonal receptors, including the RXR, that is the retinoic acid receptor. It is a ligand activated transcriptional regulatory protein. How it acts is, it interacts with retinoid X receptor and forms a heterodimeric complex, which recruits the vitamin D response elements in the target genes. This heterodimeric complex of vitamin DR, VDR and RXR, it recruits the vitamin D receptor elements in the target genes. What does then occur? This target genes activates or represses, along with the additional co-regulators, the specific cellular function by reading the DNA library and taking out the DNA encoding for specific proteins that are required for the function of the specific cell. It seems a little confusing, but we'll see in detail what is happening here. Just before we go into the cellular level, a few words about the vitamin D and tissue homeostasis. Vitamin D, through its universal VDRs, it not only takes part in the calcium metabolism and skeletal health as an endocrine action. This is the endocrine action of vitamin D from the kidneys, the regulation of calcium metabolism. That is the actual endocrine action of vitamin D. Now, there is another peripheral action also because of the through the peripheral situated vitamin D receptors, where through direct influence on cell proliferation, differentiation and apoptosis, facilitation of gene expression. This is considered as an autocrine mechanism of action of vitamin D inside the cells in the peripheral organs. This is a little about vitamin D receptor. It has two domains. First domain is DNA binding domain, which binds to the DNA, and this is the ligand binding domain, which binds to ligands like RXR and other regulators. Co-regulators are bound at this hinge area. What they do, the binding of heterodimerization with the receptors like RXR in this domain, and with the hinge area with the cofactors, they connect to the DNA binding through the DNA binding domain to the DNA, take out the encoding of the protein synthesis from the DNA, and then produce the protein that is required for the cell to function. This is how the vitamin D receptor, this is the molecular model of the vitamin D receptor, retinoid X receptor and DNA heterodimer complex. This orange color, orange is the vitamin D receptor with its DNA binding domain and ligand binding domain. With the ligand binding domain, this cyan color is of RXR, which combines, and here there is the hinge area, and this is perpendicularly attached to the DNA of this is the blue color is the DNA, which takes the genetic coding of the DNA for further action to convert it into proteins. This is the basic model of the heterodimer complex of vitamin D receptor and retinoid X receptor. This is only one example. Retinoid X receptor and vitamin D receptor has got other molecules also as heterodimer partners. This brings out an important fact to be noted, as we have already seen. This is the overview. First, we have got the endocrine part, as we have already seen from the liver, kidney, and independent of this, there is the autocrine part inside the cells, where the gene transcription is done through the heterodimerization of RXR and VDR through stimulation of VDR elements. So this is how the basic molecular action take place, with differentiation, anti-inflammatory actions, immune suppression and immune regulation, apoptosis and cell cycle arrest, as well as cell cycle. The important thing here is, once the action is over, both inside the cell as well as inside the kidney, the active vitamin D is inactivated to calcitroic acid immediately, so that the action does not continue. This is also very important, because once the action has occurred, then the active metabolite of vitamin D, that is calcitriol, is inactivated. So further action does not occur and the action is limited there. So at each and every step of this, if anything happens, disease can occur. Now, just to give a simple example of how this occurs. Imagine there is a signal or demand from a cell. The calcitriol, as we have seen, it acts as a key link in the signaling apparatus. This is the signaling apparatus of the cell that connects extracellular stimulus to genomic response in the cell. It has become clear in recent years that many tissues possess the proteins, enzymes, and signaling molecules that they need in virtual form only. In the beta cells of the, for example, beta islet cells of the pancreas, if you go there and check, you will not find insulin there. The production of insulin is encoded in the DNA blueprints in the nucleus of the beta islet cells. And this is brought out by the calcitriol and VDR complex and RXR complex. So when the cells of such tissues are exposed to an extracellular stimulus or signal that calls for them to mount a response, for example, for the pancreas beta islet cells, it will be an increase in the sugar level. That requires some of these proteins or catalysts. They do so by opening up their library of DNA blueprints, finding the ones that are appropriate for the situation, and then synthesizing those proteins by transcribing the information that is encoded in the DNA. Is it confusing? No. Of course, we know what I was told. There is DNA of each and every cell has been encoded by the information of the transcription of protein that is required for the function of that particular cell. So what our vitamin D receptor and vitamin D complex do, along with the RXR and other co-regulators, they open, it is like a key. They open the DNA library and bring out the protein transcriptase, the genomes, and then stimulate the other molecular factors to produce the proteins from the amino acid sequences. So this is what happens. The signal of, as I told you, the signal of beta islet cells in the pancreas may be increase of circulating glucose levels. Demand will be to produce insulin, and response, of course, will be the secretion of proper amounts of insulin. This is how the cell in each and every cell in each and every tissue works. All these functions are actually encoded in the DNA of beta cells of pancreatic islet cells. Calcidiol. this is the latest discovery, not latest, it has been there since more than 10 to 15 years, but latest for us who have studied the molecular biology long back. Calcidiol is a as you know, hydroxylated to calcitiol inside the cells, like in kidney. And calcitiol unlocks the DNA library along with the RXR receptor and other regulators to transcribe the endocrine production and secretion of insulin. This is in the pancreatic cell. For the molecular and cellular genetic decoding, all the cells in our body needs vitamin D and other regulators. Just note the point, all the cells, whether it is the release of antimicrobial peptides by the innate immune cells like macrophages or the production of antibodies in response, production of antibodies in response to antigen stimulation by the beta lymphocytes, or the response of muscle contraction in a signal demanding flight from the place of attack, and so on. So whatever response our body or our cells have to do, this is all managed or mediated through vitamin D and VDR receptors, as well as the other regulators and RXR. So this is actually vitamin D functions as a hormone, like all other hormones. We are only taking the example of vitamin D here. Each and we know how the other hormones, thyroid hormone, pituitary hormones, all these adrenal hormones, all this work. So all these hormones work in a similar way on their target tissues. Now it is very easy to understand that when vitamin D and other associated regulators are in optimum level like this, any stimulus will produce proper response in the cells. That is very easy to understand. So there will be normal functioning of our systems. Now imagine what will happen if the level of vitamin D is low, circulating vitamin D is low, and hence the available activated bioactive vitamin D is low inside the cell. What will happen? The stimulus will be there, but the response will be poor. The proper response will be poor. Same, to take another easy example, it is a bulb running on low voltage. The bulb will be lightened, but it will be dimly lightened. Likewise, our systems will be working in a low voltage if there is a deficiency of vitamin D and other nutrients. Now, this is not as simple as we think. It is not only vitamin D deficiency. Imagine there is a mutation of vitamin D gene. Of course, there is hereditary vitamin D resistant rickets. It's a very rare autosomal recessive disease caused by mutations in the vitamin D receptor. So in this, patients exhibit very severe rickets and hypocalcemia that is not responsive to vitamin D because of the mutation in the vitamin D gene. So vitamin D to work properly, you need a proper vitamin D receptor as well. Luckily, this is a very rare entity. Now, another problem is vitamin D receptor polymorphism and heterogeneity. We have already seen that expression and nuclear activation of vitamin D receptors are necessary for the effects of vitamin D. Several genetic variations have been identified in the VDR. These DNA sequence variations, which occur frequently in the population, are referred to as polymorphisms. These polymorphisms resulting in heterogeneity in the molecular response of calcitiol can cause or have biological effects, including autoimmune changes in the cells. So when we think about autoimmune diseases, this is another cause of autoimmune diseases. We'll see in detail the studies that have come out regarding this in the next talk. And we have a guest speaker next week who will be Dr. Renu Mathani from Pune, who will be speaking on autoimmunity and vitamin D in detail. Now, Another problem is about the vitamin D binding polymorphism. You know, vitamin D binding protein is the protein that carries activated vitamin D, as well as the vitamin D metabolites, the circulating vitamin D to the cells as well as to the target organs. So now, if the vitamin D binding protein has got polymorphism and it is not working well, that also interferes with the proper functioning of calcitriol. It is a single binding site for all vitamin D metabolites and a transport protein. It is polymorphism associated with susceptibility. It is associated with the susceptibility to a large number of chronic diseases like osteoporosis, type 1 and type 2 diabetes, thyroid autoimmunity, inflammatory bowel disease, and chronic obstructive lung disease. However, the exact role of the vitamin D binding protein in the pathophysiology of these diseases is not currently completely understood. There is some evidence that vitamin D binding protein polymorphism may be acting, but there is no evidence as such to pinpoint the exact mechanism by which this happens. Now, this is a very interesting project going on. It is launched by the UK Biotechnology and Biological Sciences Research Council, the VitaDex project. It is a as we know, it's a well-known fact that despite large amounts of vitamin D located in their adipose tissue, obese individuals are often vitamin D deficient. Obese individuals, usually in their adipose tissue and fat tissue, there is a huge amount of vitamin D deposited, but it is not available for them, for their body to properly utilize. So this is the project that is exploring the possibility of bringing out this hidden or the trapped vitamin D out. Actually, this trapping of vitamin D in the fat tissue may be due to either insufficient lipolytic stimulation or due to tissue dysfunction or adaptation resulting from adipose expansion. Too much of fat surrounding the vitamin D, and vitamin D cannot actually, you can see the picture. When it is closed, vitamin D cannot enter the circulation and act on the cells. So emerging evidence suggests that exercise may mobilize the vitamin D from adipose tissue, even in the absence of weight loss. So obese people who are habitually vitamin D deficient, they can, even without vitamin D supplement or vitamin D from the food sources, they can get sufficient amount of, this is a very interesting project. They can get sufficient amount of vitamin D by just doing regular exercise. Even if they don't lose the weight, the regular exercise brings out the vitamin D trapped inside the fat tissue. The VitaDex project will determine whether increasing physical activity via exercise represents a potentially useful strategy to mobilize vitamin D from adipose tissue of the obese. The result is expected by 2021. The classical action of vitamin D, of course, we already know. Calcitriol regulates plasma ionized calcium and phosphate levels at 3 levels. One is at the bone level, the other is through the bone synthesis, mineralization that we have seen, how the axis of parathormone, calcium, phosphates, and vitamin D acts there, and also through bone resorption. This is very important. Even in the in situations where there is calcium deficit, the bone resorption stimulated through vitamin D can maintain that calcium levels in the body. So this is very important because please mute yourself. One thing is that unless you correct the calcium levels, there is going to be bone resorption. This is what happens in osteoporosis, we all know. So this is one mechanism. I'm not going into details because this is the classical action everybody knows. This is what we have already studied long time back. The parathormone and calcitriol It stimulates calcium renal reabsorption and mobilization from bones, as well as it also regulates the absorption from the intestines. This we have already seen. Now, what are the molecular mechanisms? In the intestine, when in a low calcium diet, the absorption is through the transcellular route, through the cell. It is also regulated by the vitamin D and vitamin DR. And when there is high calcium level, it is paracellular transport. Same thing, what is happening in the renal system also. There also vitamin D and VDR complex, along with the activation of the cytochrome hydroxylase enzymes. This regulate the renal reabsorption of the calcium. Skeletal effects also during a negative calcium balance, when VDR action in the intestine is impaired or dietary calcium intake is low, intestinal calcium absorption is decreased, that we know. So now the normal serum calcium levels are maintained by increased calcitriol and parathyroid levels, which will increase bone resorption and reduce bone matrix mineralization. You might, most of you must be knowing that in 10 to 15 years, all our bones in our body are remodeled. They are reabsorbed and rebuilt. This is happening. This is a quite continuous occurrence. This is during the positive calcium balance. The normal serum calcitriol levels promote intestinal calcium absorption when there is a positive calcium balance. This pathway will usually deliver sufficient calcium for adequate bone matrix mineralization. Now, what does vitamin do to make such a difference in health? See, our genes don't want to be making proteins continuously. So vitamin D not only stimulates the genes, takes out the genetic information and stimulate them to transcribe proteins. It also has got an on and off mechanism in the body. This is achieved through regulators, and vitamin D is the major regulator in humans, with over 230 VDRs in the nuclei of most tissues. There are around 230 vitamin D receptors in most of our things. So in short, vitamin D affects gene expression by turning genes on and off. It's like a switch which turns the genes on and off. The primary function, as we have already seen, is to access information stored in the DNA and enabling them to respond to routine, ordinary signals and stimuli. So in the absence, it is well understood that in the absence of adequate vitamin D, none of our bodies and systems will work well. And it is necessary for the body to absorb enough calcium from our food, in addition to the peripheral activities. But remember, in all these actions, vitamin D is not causative, but it is enabling. It is an enabling factor. It is not a causative. When we say vitamin D, except for rickets or osteomalacia, even to an extent osteoporosis, vitamin D may be causative. But for other diseases, you cannot say vitamin D is causative. It enables necessary or optimum cellular function. When it is deficient, along with other nutrients, the cellular function will get deranged and body will go into disease. So this is what we have to understand. If, for example, vitamin D deficiency or vitamin D metabolism deficiency in action causes the dysregulation of our immune system and causes autoimmunity, and the patient who has got a genetic tendency towards psoriasis gets psoriasis, can he be cured with vitamin D? That is what we are telling. Could be to an extent. We know that calcitriol is one of the most effective, not one of the most effective, after of course our coal tar, dear old coal tar of old times, which of course must have been even working through vitamin D. Now when when we think out of it, because applying coal tar and our PUA therapy, all and now the narrowband UVB acting on the skin could be acting through the vitamin D, calcitriol. And we know the calcitriol ointments are the calcipotriol ointments are the best topical treatment for psoriasis now. So it's all, but if you give injection vitamin D in a high dose, will it cure psoriasis? No, because as I told, we discussed earlier about the machine getting out of control or getting damaged. It cannot come back. The deficiency, the damage has already been done. Damage may not be repaired, but it can repair further damage. It can prevent further damage. Once you understand this, then the mocking of vitamin D goes away. It is part of, it is a essential vitamin, which is very essential and much more essential than other vitamins. Why? Because that is the only vitamin which our body, through the evolution, not only our body, all the organisms, including plants, their photosynthesis, they produce vitamin D because it is a cellular essential necessity. But remember, vitamin D is not a standalone nutrient. I have seen many patients with vitamin D level below 5 nanogram per ml coming. Two problems. One is they have been only given vitamin D. It's not going to work. One. Second, they were told to take it for 3 months. And maybe the doctor did not tell them to stop, but most patients think that 3 months is a course. We should always tell them there is no course for vitamin D. You don't have a course for food. You take food for 1 day and say, oh, I'm satisfied for life. No. Vitamin D and other nutrients should be there throughout your life, and you should maintain a normal level to avoid getting ill through the deficiency of these essential nutrients. For vitamin D, there is a special case. It cannot work alone because, especially magnesium. Proper magnesium levels are important. Magnesium acts as a coenzyme to the hydroxylase enzymes that converts vitamin D to the active vitamin Ds. You have 5 to 8 positions in the vitamin D metabolism.
Oh, magnesium undo idinadakke.
Please turn off your audio. Thank you. So vitamin D, whenever you are giving vitamin D, make sure that the patient also gets magnesium either through the food or through the supplements. Now, vitamin A, we have already seen, they are molecular partners, the heterodimer partners, and it has been proven that vitamin D response and activity is reduced when the intake of vitamin A is below 5,000 international units per day. So vitamin D cannot work without magnesium and vitamin A. And what about vitamin K2? That is another factor. There is a synergy between vitamin A, D, and K2 for the adequate bone and teeth protection, and also protection against soft tissue calcification and adequate growth. So the list is growing. See, these are from the different reports and studies. These are the essential vital minerals: magnesium, selenium, zinc, and vitamins A, E, C, K2, riboflavin B2, methylcobalamin, omega-3 fatty acids. All these are essential for vitamin D to work properly. You know, vegans, we have got lots of vegans here. They are not taking supplements. Check their vitamin D and vitamin B12, they will be deficient. So fat diets are one cause for vitamin D deficiency, and improper unhealthy food habits are the main. So this points towards one thing only. Essentially, a well-balanced diet with a healthy lifestyle, with proper exercise, sun exposure, proper sleep, sufficient hydration. All these are essential for maintaining our health and for the proper functioning of our body. It is not just vitamin D. This is what we have to stress. Just because we are talking about vitamin D in length, it does not mean vitamin D is the only thing or vitamin D is the most important. No. This is a slide courtesy Dr. P. K. Shashidharan from us. Why vitamin D deficiency? There are many causes beautifully summarized in this picture. Increased melanin, no sunlight exposure, then no balanced diet, no vegetables and fruits, no non-veg food or supplements, lack of awareness, poverty, excess alcohol, in short, malnutrition, plus liver diseases caused by either overeating, alcohol, or toxins, kidney diseases caused by hypertension, diabetes, and toxins, hypomagnesemia. These are the main reasons for deficiency. We'll see about deficiency and the proper dosage schedule in the next talk. Now we come to one of the most important actions of vitamin D, that is in the immune system. It has come to the limelight during this pandemic of COVID-19. As you all know, cells of immune system also possess, as we have heard now, the cytochrome hydroxylation enzymes. That is cytochrome P27A1, 27B1, etc. to perform both hydroxylation steps needed to generate bioactive vitamin D, the 125 hydroxy vitamin D3 or calcitriol. As we have seen already, calcitriol binds to the VDR to induce a range of immune modulatory effects. Now, just see here in the left side, in the monocytes and macrophages. That is, those comes in the initial innate immunity. Vitamin D increases through the VDR and the stimulation by the hydroxylation enzymes. It stimulates the innate immunity. We'll see in detail the cellular mechanism in the next coming slides. And when it comes to the adaptive stage, all the dendritic cells, the effective and memory T cells and B cell, all these are regulated rather than stimulated. And this is very important, especially in infections like COVID-19, where cytokine storm is the main cause for ARDS and death. So this we have to be very just put in your mind. And once you understand this, then there is no point in telling that vitamin D does not have, there is no proof that vitamin D does not have any effect in the control of or treatment of our COVID-19 or infections like that. To take the innate immune system, as we all know, it is the first line of defense. It activates macrophages, neutrophils, and other cells to attack and destroy the attacking pathogen. Vitamin D binds to these through cells and facilitates gene expression that makes the cells better at killing the pathogens. This is how vitamin D acts in the innate thing. Not only that, vitamin D has got much more influences, right from the increase of physical barrier function, the chemotaxis, autophagy, and phagolysomal fusions, and it also induces antimicrobial peptides, we will see in the next slide, and also influences the gut microbiota, which reduces inflammation and reduce gut permeability. This is the first, even starting from the physical barrier of the innate immunity, right till the destruction of the attacking pathogens through the production of antimicrobial agents. Vitamin D is involved in this. This is a very illustrative slide. This is a macrophage. You can see the TLR. This is the toll-like receptor, which allows the immune system to recognize microbes by looking at their molecular patterns. So whatever pathogen has come, whether it is a Mycobacterium tuberculosis or COVID-19 coronavirus to the TLR recognizes, binds to them, and then sends signals to the cell. Intracellular signals are sent. So we see when the pathogen invades the cell, the toll-like receptor signaling is activated with increased expression of vitamin D receptors and the cytochrome here. This is increased, stimulated, there is increased expression, and there is hydroxylation through these enzymes of the inactive vitamin D, circulating vitamin D, which has entered the cell to this response, and then it becomes the bioactive calcitriol. That is the the third step. And in the fourth step, calcitriol binds with vitamin D. Once it is bound with, along with the RXR and other thing, it goes to the nucleus, binds, forms the heterodimer with the DNA, and then stimulates the expression, the nucleus, from the DNA library. It takes out the information to produce the antimicrobial peptide, CAMP, that is called antimicrobial peptide, the cathelicidin as well as defensin. Once these are produced, they enter into the phagosomes. The phagosomes has already engulfed the pathogen and then help the phagosomes to engulf and destroy or kill the virus or bacteria, whatever the cases may be. See how important it is. It is not only in macrophages, monocytes, in the T lymphocytes and B lymphocytes also, this mechanism is going on. Whichever pathogen enters, it is this mechanism that produces the antimicrobial attack of the innate immune system. So imagine what happens if this is not working well. If the vitamin D deficiency is there, what will happen? For example, in COVID, the virus enters, there is no cathelicidin and defensin to destroy it. It multiplies. There is increased viral multiplication. There is increased viral load, and this viral load attracts more cytokines, and there is cytokine storm. So if the innate system is not working well, it will also lead to cytokine storm. This is a very important thing that we have to remember. This now to the adaptive immune system. We've seen the second line of defense against pathogens, and when the cells of the adaptive immune system, as we know, is B cells and T cells. They produce antibodies that protect our body from specific invaders. So what happens is, once the vitamin D, actually it has got a calming effect. In the innate system, it has got a stimulatory effect, but in the adaptive immune system, it has got a calming or regulatory effect, where it suppresses the undue cytokine levels and inflammatory agents like interferon or the tumor necrosing factors. So this actually decreases the inflammation and the chances of the increased hyper responsiveness of the immune system. So this is also one important factor that could actually control the cytokine storm in infections like this is the, I'm not going into details, how each and every step of the adaptive inflammation is being controlled or regulated by sufficient amounts of vitamin D in the system and properly functioning VDRs, which suppresses the inflammatory reaction, hyperinflammatory reaction in during the attack of by the pathogens. Now, when we think about this, also see what is happening in cytokine storm. In the when we intervene in the early stages of COVID-19 pneumonia to control this cytokine storm, in the early stages, once the reaction, the cytokine storm has been triggered, it's very difficult. We'll have to go for the other methods to suppress the interleukins. So that is different, JAK inhibitors and all those things we have to bring into action. But vitamin D has got a very big, very huge role in the prevention of this cytokine storm. Another surprising finding is about the anti-aging property of vitamin D. As you can see, vitamin D stimulates the autophagy and suppresses all the activities that stimulates aging, like mitochondrial dysfunction, oxidative stress, increased inflammatory reactions, calcium signaling, epigenetics. Epigenetics means our genes changing their characteristics because DNA mutation, because of external influences from the environment, our food habits, stress, and other lifestyle habits. And other DNA disorders and telomere shortening, which all add to aging, and vitamin D suppresses all these actions, but stimulates the autophagy, which actually suppresses aging process. There is another mechanism also. Vitamin D does not act directly on this. It actually acts through the alpha-klotho, a renal hormone which has been proven as an anti-aging enzyme or core receptor that protects against skin atrophy, osteopenia, hyperphosphatemia, endothelial dysfunction, cognitive defects, and neurodegenerative disorders, and impaired hearing. Together, the calcitriol and alpha-klotho maintain the molecular signaling systems that promote growth and development and through antioxidation and homeostasis in tissues. So this is the mechanism by which they act. You can see there are two steps in this. One is along with klotho, all this which is under the control of the hormones and the kidney, the hydroxylase enzymes. The calcitriol along with klotho works not only as an anti-aging, it has got an inverse relationship with the renin-angiotensin. This we have seen in a little detail in the first talk also. 1,25 dihydroxy vitamin D or calcitriol actually suppresses the renin-angiotensin system and reduces the production of angiotensin 2, which is a vasoconstrictor inflammatory molecule producing the ACE. And it also suppresses the production of angiotensin 2, suppresses the production of ACE2, which is actually anti-inflammatory. The calcitriol is stimulatory on the ACE2. So these are the two mechanisms by which vitamin D in the kidney itself acts on the anti-aging mechanism as well as the renin-angiotensin system. This also involves the, the ACE2 receptors are the site where the proteins, same proteins of the our COVID-19 attaches to the cell, and this actually deactivates the ACE2 and gives place to this inflammatory and actually stimulates the inflammatory response within the cell. So vitamin D has an action there also. We'll see a little fast. This is what I told you, how we all know this, the ACE inhibitors, how the COVID-19 enters the cell. Go a little fast. Spike proteins of COVID-19 inactivates ACE2. There have been studies proving that, and ACE2 is actually the key where the COVID-19 enters the cell, but during the entrance, the ACE2 is inactivated, and that produces that stimulates the inflammatory cascade. We all know about the renin-angiotensin system. It is important in maintaining blood pressure, fluid, and salt balance. Angiotensin 2, as I told, is inflammatory in nature, and its levels are linked to disease severity. especially in COVID-19. ACE2 is the good, the beneficial molecule in there, which is actually deactivated by the COVID-19, and ACE is the deleterious one. The good ACE2 or from the angiotensin 2, once it is suppressed, this suppression is done by vitamin D calcitriol also. ACE2 is produced, which has got anti-inflammatory, anti-fibrotic, and vasodilatory action. This is how vitamin D interferes with the renin-angiotensin. It stimulates the ACE2, which has been inhibited by the SARS-CoV-2, and it inhibits ACE, which causes one of the causes of the severe COVID-19 disease. The same thing that we have already seen. Now, a few words about sources of vitamin D. The vitamin D deficiency, the sources, everything, we will discuss in the next thing. We are moving towards the last few slides. As you all know, sunlight is the best source of vitamin D, but many people can't get enough for various reasons. It could be cultural, it could be just idleness, or it can be habitual. Theoretically, but remember that we get 80% of our vitamin D from sunlight. Food and supplements that are high in vitamin D include, we have already seen cod liver oil, fatty fish, egg yolks, and mushrooms that are grown in the sunlight. This is some of the fortified as well as the natural vitamin D sources. Now, how do we know that we have vitamin D deficiency? The blood tests are the only way to know if you are vitamin D deficient. Of course, having rickets and all is there, but it is a very late occurrence. Severely deficient, we say when it is less than 10 nanogram per ml. And deficient when levels are less than 20 nanogram per ml. And insufficient between 20 to 30. And sufficient levels between 30 to 100 nanogram. Optimum levels, we can say, is around 40 to 60 nanogram per ml. Previously, it was thought that 30 nanogram is enough. But this 30 nanogram per ml as the upper level, that the optimum level that is required for vitamin D sufficiency was, brought into existence long time back when we were thinking that vitamin D had only role with bone mineralization and calcium metabolism. Now we know it is not so. Each and every cell requires vitamin D. So how can all the cells get sufficient vitamin D with only 30? So even 30 nanogram may be deficient for many people. We don't know. We need more studies about that. Because when we say that even with sufficient amount of vitamin D, some people get some diseases that may be connected, and some people get immune deficient, or some people's system does not respond to the response in a degraded way towards the pathogens. So maybe this is the cause, that actually the normal level that we are thinking of now is not actually the exact normal level. Toxic, of course, above 150 nanogram per ml. Toxicity is very rare. We'll see in the next talk in detail about this. This is just for you to get ready for the next talk. This is the optimum levels, 40 to 60. Usually 40 to 50. I would advise, well, I keep mine around 50. There are a few reports which says it is after 50, a few people have developed hypercalcemia. So to be on the safer side, let us say 40 to 50 is the optimum level. Up to 60 you can go, because many studies have shown that the toxicity of vitamin D is very rare, and it occurs with very high levels of vitamin D supplement for a prolonged period, and raising the levels to above 150 nanogram per ml. So these are all mainly case reports are very less, but there are also a few case reports showing that very low level and very little higher level of vitamin D is also bad for some diseases. So that we'll see in the next talk. So let us keep it optimum in this region. So please check your vitamin D. Most of the people, some doctors even say everybody is vitamin deficient. So why do you need to check your vitamin D? Just take vitamin supplement. I don't agree with that, because vitamin D supplement, when it is very severely deficient, like we are seeing here, severely deficient and deficient, they need more dosage to start with, and they may require some more time to become sufficient to get the sufficient dosage, to get the optimal dosage. So it is always better to do an initial test, and if it is very low, after taking the vitamin D in proper doses that we will see in the next talk, after 6 months we will have to again check. If the vitamin D is normal or this thing, you can continue with a maintenance level. And if you are regularly exposing to sunlight and taking sufficient amounts of food sources, then there is no need for vitamin supplements in people who have got normal levels. So this is the advice that we can give to patients. But remember one thing, we all know, especially those who have got decades of experience, when somebody comes with one deficiency, it is mostly due to, there will be other deficiencies as well. One. Second, the deficiency is due to their unhealthy lifestyle. Whatever we do, whatever we tell, even if we frighten them, they will go back to their old lifestyle. So this is why we say it is very easy to advise people to lead a healthy lifestyle. They always go back to their old habits again, and they go into deficiency again. As earlier what I told, some patients come and say, 3 months I took the course of vitamin D. There is no course for vitamin D or other nutrients. They have to take continuously and keep in the optimum level. Some labs report nanomole per liter. It is 1 nanogram into 2.5 is the nanomole per liter. So average, when you see here, deficiency is below 75 nanomole per liter or 30 nanogram per liter, and that is the deficiency state. And in very severely deficient, below 25 nanomole, and deficiency in below 50. This is the range, just so that you can remember these ranges. Just again to remember the insufficiency levels, 20 nanogram is equivalent to into 2.5, 50 nanomole per liter. Now, remember this is for their next talk. Rickets is just the tip of vitamin D deficiency. Nowadays, we don't see rickets, but vitamin D deficiency is very rampant. And many people say, I have got lots of vitamin D deficiency, but I don't have any of these diseases. But don't be so sure. It adds on. It is not, as we already seen. Vitamin D is not the actual cause. It is only the facilitating factor in our health, along with the other nutrients. So it is always safe to keep the vitamin D levels in the optimum level, along with other vitamins, and lead a healthy lifestyle. There are ethnic differences also. Of course, we know there are many studies where the northern Inuit natives and Amerindian aborigines, they have got very low vitamin D, but they show no signs of rickets. This has been, in many studies, this has been shown as a proof that vitamin D deficiency is not a big health issue at all. But there are many problems here. One is through evolution. When we are away from the latitude, when your sun exposure is low, or the sun rays are less throughout the year, or if your skin color is very dark, these people have, throughout the evolution, they have developed mechanisms to have normal calcium levels independent of vitamin D. There are other alternative pathways for calcium. This cannot be done if, for example, for one black African to go and live in the Arctic and Antarctic. It will be very difficult for him to adjust, because it is very difficult for him to take, unless he takes, of course, the full panel of the supplements. So it has been going on since so many years. This is why one reason how the colors have developed, the Caucasian colors, the white color, the brown color, the black color, all these are connected with it's a balance between sufficient vitamin D and the protection against cancer. For black skin people, they have to expose for more time to get sufficient vitamin D, but they should not get cancer. So skin cancer. What during the evolution, what has happened? Their skin's melanin pigment has increased to such a high degree that even if they're exposed to sunlight for a longer time, they are not prone to get skin cancers. So this is what has occurred during the evolution, the evolutionary survival mechanism. The sun exposure and vitamin D, I have already told you. It is around, depending upon your skin color. You are very fair, sun exposure between 10 a.m. to 3 p.m. from 5 to 10 minutes or 15 minutes is enough. But as your skin color darkens, so should your time of exposure increase. For brown skin people like Indians and Asians, we have to expose to half an hour to maximum 45 minutes. For dark skinned in Africa, they have to expose from 45 minutes to 1 hour. Also between 10 a.m. to 3 p.m. And it should be done to the surface area of the skin to be exposed is around 20 to 40% of the surface area of the skin. It is very difficult for modern men to do it. And imagine why we have survived through millennium, because we are not sitting in offices, air-conditioned offices without sun exposure. But we go like this, why we should think once again, why the cancer rates are increasing, why the heart disease rates are increasing, why the infectious diseases, when the pandemic came, why we were all taken to ransom. Why is it happening? It's all because of our lifestyle. So let us rethink and reschedule our lifestyle and think very seriously about how we manage our nutrition and lifestyle. There is enhanced sun exposure advice coming from the experts, that the amount of body surface area that is exposed to the sun strongly influences the vitamin D production. So what they are advising is there should be short, frequent sun exposure with as much as skin exposed as is possible under the circumstances. Instead of continuously exposing for half an hour or 45 minutes, you can take short intervals of exposure. That will actually be better, and it will also reduce the chances of not only cancer, skin aging. Of course, you know, the longer you expose to sunlight is one of the causes for wrinkling and other problems of skin aging. Now, we should also remember, this is why when patients come and say, I exposed to sunlight in early morning and in the during early evening for vitamin D. No, they may not get vitamin D, but there are non-vitamin D benefits also to sun exposure. One, even if you are taking supplement, if you expose to sunlight even in the morning or evening, that heat itself promotes the utilization or activation of vitamin D in the body. So that is one. Second, more interesting is the exposure to ultra UVR results in the production of heme oxygenase, which causes the breakdown of heme and which catalyzes the production of carbon monoxide, iron, and biliverdin. And this pathway is very important in obese people and also in cardiometabolic dysfunction. There is nitric oxide stores in the skin. This is also released during the exposure to sunlight. So nitric oxide coming to the system and causing vasodilation, will decrease the blood pressure. And also, we all know the nitric oxide is okay, even the erectile dysfunction is controlled. When in the skin and eye, when skin and eye are exposed to the sunlight, we all know that there is release of alpha melanocyte stimulating hormone, that is the MSH hormone, which activates the pro-opiomelanocortins, which are responsive neurons in the acoustic nucleus of the hypothalamus, and this could again hypothetically reduce appetite and food intake. Very important point for obese people. So part of treatment of obesity is optimum sun exposure as well. So this is the extra vitamin D benefits of sun exposure. So never discourage sun exposure for anyone. In fact, we should promote sun exposure and exercise in sun exposure as far as possible. We come to the end of part 2. Before we end, I'll just give a preview of part 3. So please don't miss the part 3 also, because you are not going to get this in any textbooks. I've seen, I had a discussion with one of our friends who told it is about vitamin D. There is not much mention in our classic or standard text. So that means vitamin D may not be that important. We cannot be so wrong. See, calcitriol, there are so many hundreds of studies linking vitamin D deficiency to many diseases, osteoporosis, of course, cancer, multiple sclerosis, the other autoimmune diseases, hypertension, heart disease, diabetes, sun damage, of course, we know, neurological disease, psoriasis, infections, all this. Are these all points to that same question that I asked earlier in the last thing also? Is vitamin D the modern snake oil? It cures all? No, sir. It will not cure everything, but definitely its optimum level may have a role in preventing many of these diseases. So let us err on the positive side. Instead of telling patients there is no effect, there is no proof, or there is no evidence. As we discussed in the last talk, evidence-based medicine may misguide us sometimes, because for everything evidence is not possible. We will see. Remember this, in all the actions, vitamin D is not causative, rather it is enabling. So with this in mind, we will go to next week for the vitamin D in diseases, and also discuss in detail the vitamin D recommendations. That is very important, because there are so many factors affecting the recommendations. So we have, as I told you, we have a guest speaker, Dr. Redu Mahatani from Pune, who has done lots of studies on vitamin D and autoimmunity, and Dr. Sunny Kurian, our founder, secretary general, and past president of I can give rights. We will meet next week, same time. So with this, I stop. Now we will take questions. Dr. Sri Kumar and Dr. Jabal, please take over for the questions session. Thank you.